at this point, let's switch our gears to two-thread inheritance. So now we are not looking at one feature anymore. We are looking at two features. For instance, we can look at the color of the seeds and the shape of the seeds. In this particular instance on this slide, we are looking at the color of the pods and the size of the stem. Are the stems tall and the pods green? Are the stems short and the pods yellow? So we're looking at two features here. And remember, in the context of Mendel, he always started with true breeding parents. In other words, a true breeding parent in this particular instance here would be tall and with green pods. From one generation to another generation to another generation, there would always be tall stems with green pods. By the same token, the other parent would be short stems with yellow pods generation after generation after generation, right? From that perspective, he knows that you have homozygous dominant for the trait in the green and tall parent and homozygous recessive for short and yellow, right? So from there, he can deduct what kind of gametes are being produced. So the gametes for the tall and green true breeding parent is going to be capital T, capital G, while the other parent is going to produce gametes that are lowercase t and lowercase g, right? Now, in the F1 generation, he observed that all individuals were tall with green pods. In other words, he had double hybrid, not only hybrid for the height, but also hybrid for the color. That's double hybrid. Now, when he did a double cross, meaning crossing a double hybrid with one parent double hybrid, another parent double hybrid, what did he observe? So what he observed was surprising to him in the sense that after looking at all the possible gametes, right? Capital T, capital G, capital T, lowercase g, and then lowercase t, capital G, lowercase t, lowercase g. He did that for the female plant and for the male plant. And by the way, how did he produce all these gametes, or how did he deduct the production of all these gametes? Let me show you how he did. So the individual, say, is capital capital T, lowercase t, and capital G, lowercase g, right? That would be a D hybrid. So for producing the gametes, to find out actually the possible gametes that it can produce, say we have this capital T, right? This capital T can end up in the same cell as the capital G. So that's one possibility. The same capital T can end up in the same cell as lowercase g. Now, that's the first focus, capital T. At this point, we are going to focus on lowercase t. So lowercase t can end up in the same cell as capital G, and lowercase t can end up in the same cell as lowercase g. This is why we see capital T, lowercase capital G, capital T, lowercase g, lowercase t, capital G, and lowercase t, lowercase g, right? So having said that, now we need to cross gametes and eggs, right? By doing so, we obtain the different genotypes here, and from the genotypes, we deduct the phenotypes, right? And having done all of that, what Mendel observed is that in the F2 generation, we had 9 plants that were tall with green pods, 3 were tall plants with yellow pods, 3 were short plants with green pods, and only 1 was short plant with yellow pods. Now, altogether, you had the possibility of 16 different types of plants, right? Out of the 16, you have the distribution 9, 3, 3, 1. Okay, that's the case of the double hybrid. The conclusions, he said that, well, when considering 2 features, actually the 2 features can be carried by 2 separate chromosomes. In other words, if you consider among homologous chromosomes pair number 1 and pair number 2, and you are looking at 2 features, the height of the stem and the color of the pods, right? The height of the stem can be carried by homologous chromosome number 1, right? While the shape, the color of the seeds, the color of the pods can be carried by set of chromosome number 2. Considering that, he said that he reached a conclusion that these chromosomes that are obviously different can separate independently of each other. So they don't depend on each other as to how they are going to separate during meiosis. This is the law of independent assortment. That law of independent assortment explains the genetic diversity that we see in the population. So at this point, we have 2 laws. The first law is that the 2 alleles have to end up in 2 separate cells. That's a law of segregation, right? The second law at this point is the law of independent assortment. Chromosomes separate in different cells regardless of each other, okay? So each pair of factor segregate independently of other pairs, right? Which throws in the possibility of combinations, right? Which are going to cause the production of a wide diversity of gametes. Now, at this point, we have not even started looking into how crossing overs can affect the genetic diversity, which is another story, because that story is going to make things more complex. All right, so just like the 1-thread analysis, he can also do the 2-thread test cross, where he would start with a true breeding for the recessive features and an unknown breeding for the dominant feature. Are they dominant homozygous? Are they heterozygous? He doesn't know, right? So here we have flies, fruit flies, and these flies are going to be one with long wings and gray body. The other one would have black body and short wings, right? Black and short being recessive over gray and long wings, right? So what he did is to actually cross these two individuals, okay? By crossing these two individuals, if he observes that you have one possibility of each, in other words, you have one individual with long wings and gray body, okay, which is the parental features here. Another individual with long wings and black body, that's a mix, right? Another individual with short wings and gray body, that's another mix, right? And then finally, an individual with short wings and black body, that's a parental feature here. So this would be, in the case, the unknown individual would be double hybrid. That would be the outcome, all right? But if the individual was completely dominant, okay, every single individual would actually have long wings and gray body, okay? That would be the outcome if the parent was a true breeding for color of the body, gray, and length of the wings, long, okay? So now let's take a look at meiosis from the perspective of Mendel's laws. So we are starting with two pairs of chromosomes, right? This is one pair and this is another pair. And let's see what happens with the two pairs of chromosomes during meiosis, right? So during meiosis, first and foremost, the chromosomes will have to be duplicated during the interphase, okay? This is what you see here, right? Now, as you see that, now during prophase, we're going to have, let's not focus on crossing over here. We're going to see a condensation of the chromosomes, and eventually from prophase, we're going to go into metaphase 1. From prophase 1, we're going to enter into metaphase 1. During metaphase 1, the chromosomes are going to line up on the equator line by pair, right? Now, because they line up by pair, the way pair A is going to line up is not the same way pair B is going to line up, right? So that we have two possibilities. We have this possibilities of line up, and we have this possibilities of line up, okay? These two possibilities are going to boil down to the type of gametes we are going to obtain at the end of meiosis 2, right? So at the end of the day, we are going to have the following possibilities. Some gametes being capital B, capital A, capital B. Some being lowercase a, lowercase b. Some lowercase a, capital B. Some capital A, lowercase b. These are all the possibilities that exist for this parent cell as initial cell, right? These are the possibilities of gametes, okay? So considering this, the role of meiosis, we notice that some features are dominant, some features are recessive. However, what we observe when it comes to, looking at details in a population, we see that not all dominant alleles are necessarily normal. Now, the reason why I mention this, it's because there is a tendency to believe that dominant means normal, recessive means abnormal. That is not true at all, okay? That is not true. You want to know that some alleles represent a gain of function, some allele represent a loss of function. That gain of function can be abnormal. It can be normal too, right? I mean, let me correct things. That gain of function can be abnormal. The loss of function can be actually that the function normally is never there at the first place, right? And that when you gain the function, now you are going to have a feature that you didn't have. By the same token, you can have a function in a normal condition, and a mutation is going to cause a loss of that function, which is going to cause you to lose something, right? But remember that not all dominant alleles are normal. An example is that polydactyly, which is an extra finger in the hands or in the toes or both, right, is actually a dominant feature. So polydactyly means several fingers, right? It's a dominant feature. Now let's take a look at some pedigree. What do we call a pedigree? A pedigree is actually a graphic representation of the relationship between parent and offspring, right? By looking at that relationship, we can actually track down how features are being passed from parents to offspring, right? Are these features recessive? Are they dominant? Typically, when we look at the pedigree, and I'm not going to bug you with the details, but just know that in a pedigree, a square represents males, a circle represents females. That's one thing you want to know. The second thing is that an empty square represents normal male, unaffected male. A square that is filled with color represents affected males. By the same token, a circle that is empty represents unaffected female. A circle that is filled with color represents affected female, right? Now, because this is life on planet Earth, I'm not going to bug you with the details of that. But what I want you to know is that in a recessive pedigree, what we can see is that here we have a female that is affected, that is actually marrying a male that is unaffected, but we don't know if that male is homozygous dominant or heterozygous. We don't know that, but we do know that if the feature is caused by a recessive allele, then and that mom is affected, that means that mom is homozygous recessive, right? So in that condition, well, we see from the history of this family that the offspring right here are unaffected. Okay, the offspring are unaffected. Know that mom, as the parent here, can only produce what kind of gamete? Dad, we know that he can produce at least one kind of gamete, if not two, right? But what we do see anyway is that in this particular example, the children, which are both female, are unaffected, right? However, these children are heterozygous, okay? Now, when we look at the offspring of the children, right, we see that they have female here, unaffected, and this individual here from the second generation has many children. None of them is affected. However, what we do observe is that in the third generation, right, when this individual has children with this individual right here, we have that many children are affected. Okay? Now, what we do observe in autosomal recessive pedigree, now autosomal, these are the chromosomes that are not associated with sex, right? These are chromosomes number 1 through 22, okay? Autosomal recessive disorders most of the time skip a generation, at least skip one generation. But we can see that in this particular instance, they skip 2 generations, right? The third generation, only in the fourth generation, we see individuals that are affected. So affected children can have unaffected parents. That one thing that is very important to know. And that heterozygote individual have a normal phenotype. Both male and female can be equally affected or unaffected. So sex is not involved in here. Just like we have autosomal recessive pedigrees, we can also have autosomal dominant pedigrees where the dominant allele is being expressed. When the dominant allele is being expressed, anytime that dominant allele is present, it's going to be expressed. In general, as rule of thumb, dominant pedigrees most of the time do not skip a generation, right? From one generation to another, we are going to see individuals that are affected. This is what we see here, right? Parents are both affected. Then they're going to have offspring that are affected. And the offspring is going to have also the next generation of children that are affected. Okay, so there is no skipping of a generation. That speaks to how strong that allele is being expressed in the population. So affected children will have at least one affected parent. Heterozygous children are going to be affected, and both males and female are equally affected in autosomal dominant pedigree, right? No skipping of a generation. All right, so let's take a look at some disease of interest to us. We have what we call methemoglobinemia. Methemoglobinemia, it's a condition where hemoglobin is actually abnormal. Now, hemoglobin, it's the protein in red blood cell that is responsible for carrying oxygen, right? That methemoglobin is some version of modified hemoglobin that is not able to actually capture and release oxygen normally. And this is happening because of one particular enzyme that is not present in this individual. This enzyme is the deaphorase. That deaphorase is technically responsible for converting the hemoglobin to a normal hemoglobin. By that enzyme being there, that hemoglobin is not normal, so that the individual is actually going to have to be bluish like this, okay? And this can lead to a situation of hypoxia, low oxygen. And that low oxygen can be a major problem. You know that oxygen plays a very important role in ATP production, right? Because oxygen is involved in cellular respiration, which is basically the process by which we produce most of our ATP, right? And we know that ATP fuels all the forms of cellular work that are happening in our system. So hypoxia that is caused by methaglobinemia can lead to very bad consequences, right? You have a normal individual here with a normal hemoglobin that is red, while this hemoglobin here is not red, it's actually dark or blue, if you wish. That doesn't mean that it's color blue, but it's not bright red, basically. So that's one recessive disorder, methaglobinemia. Another condition, it's cystic fibrosis. That cystic fibrosis, it's actually a condition in which the channel that is a transmembrane channel designed to transport chloride ions across the plasma membrane is not allowing the chloride ion to cross properly across the plasma membrane. Because of the lack of functionality of this abnormal channel, what happens is that chloride is concentrated on one side, keeping on that side water with it, because remember that some chemicals are not able to cross the plasma membrane. Because they are not able to cross the plasma membrane, we have osmosis, right? And now, because of a high concentration of chloride on this side of the membrane, there is a need for water there, so that water is going to stay inside of the cell. While outside, you are going to have a huge amount of mucus. That mucus is going to be thick, right? That mucus is going to be thick and capture about anything. That's why cystic fibrosis individuals are prone to infections, including respiratory infection, digestive tract infections, and infections of most areas where we have mucous membranes, right? So that is basically the situation in cystic fibrosis individual, which is a recessive disorder, which means that you need to receive a defective gene from mom and a defective gene from dad. This is also the case for methaglobinemia. Another disorder is alkaptonuria. Alkaptonuria, it's when our body is not able to properly process a particular amino acid in our system, that is phenylalanine, right? There is also another amino acid that we cannot process properly. I cannot remember exactly the name of the amino acid, but bottom line, what we actually see, because of that lack of enzyme and lack of proper processing of phenylalanine, we have a dark precipitate that forms and that stain the tissue, like you see here on the picture, right? That enzyme that is not present is actually homogentisate oxygenase. Okay, that homogentisate oxygenase is actually causing, the absence of it is causing the accumulation of this product that is actually dark. Okay, while that product should be broken down in the normal individual. Finally, the last recessive trait I would like to address, it's the sickle cell trait. In the sickle cell trait, we have that the hemoglobin is abnormally shaped. That hemoglobin that is abnormally shaped is causing the cells to actually look sickled, red blood cells specifically, while the normal cells are actually biconcave, right? If we were to represent the cell at another angle, the cells would actually look like this. Okay? Biconcave, that would be the look of the cells, while the abnormal cells are basically the shape of a cycle like this. Okay? This this production of sickle cell are actually going to be unable to properly negotiate the small capillaries, which is going to cause that the capillaries walls are going to be hurt. But very importantly, there is not going to be proper flow of the blood in organs, causing all kind of issues.